studying a deadly type of breast cancer called triple-negative. Johns Hopkins medicine scientists say they have identified key molecular differences between cancer cells that cling to an initial tumor and those that venture off to form distant tumors. The research, using mouse models and human tissues, could pave the way for developing new treatments that target such molecular variations. A report on the findings is published August 3 in Science Translational Medicine. We have long needed new treatment targets and options for triple-negative breast cancers, says Andrew Ewald, Ph.D., the Virginia D. Settis Professor in Basic Science Research and Director, Department of Cell Biology at the Johns Hopkins University School of Medicine and co-leader of the Cancer Eye. These cancers often return within 3 years of diagnosis, and treatments used for other breast cancers don't typically work for triple-negative. An estimated 10% to 20% of the 280,000 breast cancers diagnosed in the U.S. each year are triple-negative, and the rate is higher among African America. The lethal nature of this type of cancer is marked by the fact that its cells lack molecular flags on its surface that connect with the hormones estrogen and progesterone and a cancer growth-promoting protein called HER2/neu. For the current study, the research team looked at molecular differences between initial, or primary, triple-negative breast cancer sites and areas where it spread, or metastatic sites, among 3 different types of cells: mouse models, human cancers implanted into mice and samples of both primary. The researchers used a combination of machine learning, cellular imaging, and biochemical analysis to identify differences in the genetic expression patterns of initial and metastatic tumors. The bad news from our study is that cells from metastatic sites are super-optimized for migration and resisting treatment, says Ewald. The good news is that we identified several proteins called transcription factors that these cells require to handle the challenges of migrating and thriving at metastatic sites, and we may be able to design new therapies that target these transcription factors. More specifically, Ewald and Johns. The scientists found that when triple-negative breast cancer cells invade other tissues on their way to another part of the body, they gain 2 cellular properties: better movement and survival. To look more closely at molecules involved in hybrid EMT states, the scientists sought the help of Alana Fertig, Ph.D., Division Director and Associate Director of Quantitative Sciences and Co-Director of the Convergence Institute at the Johns Hopkins Kimmel Cancer Center, to track the molecular P. The scientists found that most of the metastatic cells morph into the more mobile, better surviving, hybrid EMT state. At the molecular level, the most metastatic cells produced 5 proteins called transcription factors: GRHL2, FOX2, ZEB1, ZEB2 and OVAL1, that foster the making of proteins involved in either cancer cell invasion or colony formation. His team is studying ways to block the transcription factors genes or their resulting proteins to halt metastatic cancer growth, as well as whether the same molecular and cellular changes happen in other cancers, such as those in the colon, adrenal glands, stomach, and small intestine. The research was supported by the Breast Cancer Research Foundation, the Twisted Pink Foundation, Hope Scarves, the Jane Kaskina's Ted Giovanni's Foundation, the Cindy Rosencrans Fund for Triple-Negative Breast Cancer Research and the National Institutes of Health's National Cancer Institute, U01CA217. Johns Hopkins Medicine. 2022, August 8. Scientists identify novel molecular biomarkers in cells that spread a deadly form of breast cancer. Science Daily. Retrieved August 8, 2022 from ift.tt/Q6-PILC. Johns Hopkins Medicine. Scientists identify novel molecular biomarkers in cells that spread a deadly form of breast cancer. Science Daily. https://ift. TT, Q6PILKF, accessed August 8, 2022. Thanks for watching. Please subscribe my channel for more videos.